slowly start in and then allow people to join in as we go. But welcome to the Atheronet Journal Club. We are very excited today to have and welcome Dr. Chad Weldy, who's a cardiologist and an instructor at Stanford Center for Inherited Cardiovascular Disease. So Chad has a very interesting background because, as he was previously saying, he has a basic research background with a PhD and a postdoctoral experience from Washington University. And then he went on to pursue an MD at Duke University with a postdoctoral fellowship at Stanford University at the group of Thomas Kortormos. So Chad has an experience with epigenetics and myocardiopathies. And then today he's going to present the latest research that's been published in Nature Cardiovascular Research on the role of smooth muscle cell expression of RNA editing enzyme ADAR1 and its control and the activation of RNA sensor MDA5 in atherosclerosis. Thank you so much, Chad, for agreeing to be part of our JCs, and we're very happy to hear the presentation.
Yeah, thanks for the introduction and the invitation. I think this will be a great chance for us to be able to talk about what I think is a fun and exciting story. And hopefully, if there's questions, we can talk more at the end as well. Um, but I think first, the way I wanted to start this discussion was by presenting a conceptual framework of how we think about complex genetics in complex disease. And I think ultimately we can think of it as that human disease is driven by transcriptional programs that influence cellular state and function. We can even look at this on single cell resolution to understand how an individual cell may have changes in gene expression. And ultimately, when we think about common genetic variation, we're oftentimes thinking about this in the context of it influencing the expression of a gene. And so, of course, we can think of this as an eQTL or an expression quantitative trait loci. And the goal of studies such as this, such as genome-wide association studies, is that we can unmask crucial mechanisms of disease by understanding how gene variants modify gene expression, and then we can use this to be leveraged to discover novel clinical therapies. But I would argue that, particularly, say, if we take coronary artery disease, now despite over 300 genetic loci associated with coronary disease, the promise of these therapies to actually lead to actual new clinical therapy outside of lipid lowering has truly not been fulfilled. And I would make an argument that this eQTL framework is perhaps an incomplete lens, that we can understand that gene variants can modify expression. Of course, that's important. But we also know that gene variants can modify, for example, RNA splicing, and we would call these splicing QTL variants. And RNA itself is modified in a lot of different ways. So, for example, A to I RNA editing, which is something I'm going to be talking about a lot today, as well as M6A methylation. These are also variants that can have changes in RNA itself that can be influenced by the genetic variation. And so I want to pose this question of that, are we underappreciating how variants impact RNA modifications and actually influence disease risk? So if I dig into ADAR-mediated RNA editing for a moment, I think this is a very intriguing yet entirely underappreciated area of cellular biology, where ADAR enzyme, this adenosine deaminase acting on RNA, can bind to double-stranded RNA and facilitate the deamination of adenosine into inosine. And so this A to I edit, if this occurs in a coding region of coding sequence of an RNA molecule, it can actually just change the amino acid sequence. But intriguingly, A to I RNA editing is highly frequent. There are thousands of RNA editing sites across the genome with highly frequent RNA editing. And actually, the majority in mammalian cells, the majority of RNA editing actually occurs in non-coding regions of RNA. And so it might be a question, why is it that we would have all these non-coding regions of RNA getting edited? Well, it turns out there's this intriguing quirk in our biology, which is that in normal RNA transcription, the long repetitive sequences at the ends of RNA molecules actually fold over and make these long double-stranded RNA structures. And these double-stranded RNA structures can actually mimic a double-stranded RNA virus. And we have mechanisms to be able to sense what would be thought of as a double-strand RNA virus. And so, for example, MDA5 is a key endogenous double-strand RNA sensor. And when it senses this, it triggers what would effectively an innate immune mediator response. And a specific response we call an interferon stimulated gene response or ISG response. Now, the reason why our cells are not constantly activating this ISG response is that ADAR1 can actually bind to these double-strand RNA, and by editing, it actually changes the structure of these double-strand RNA molecules. And when it does so, it basically evades MDA5 from sensing this RNA, and you no longer have this ISG activation. And so it was mentioned earlier, I'm a clinician. I'm someone who I oftentimes care for patients more with rare and ultra rare diseases. And I like to think of it as that we can learn a lot about our biology through the lens of rare genetics in our clinical patients. And I think an intriguing example of this within ADAR biology is that if you have a loss of function of ADAR1, patients develop a severe pediatric autoinflammatory disorder called Aicardi-Goutieres syndrome. There's also a separate syndrome called Singleton-Merten syndrome that presents a little bit later and has a little bit different phenotype, but similarly has been reported with loss of function variants in ADAR1. And so what it's highlighting this idea that if you lose ADAR1, it's implicating this activation of MDA5 as a driver of this disease. And intriguingly, if there are actually rare gain of function variants of MDA5 that similarly result in both Aicardi-Goutieres syndrome as well as Singleton-Merten syndrome. And so it's painting this axis of ADAR1 to double-strand RNA and MDA5, and the downstream inflammatory signaling is causing both a human disease and a rare disease context. Um, in mouse, if you say delete the deaminase function of ADAR and this mouse model, this E86A, 861A variant, the mice are actually embryonic lethal by E13.5. But intriguingly, if you delete MDA5, the gene symbol of which is IFIH1, if you delete IFIH1, then it's actually a rescued phenotype and these mice can live until adulthood. And so it's highlighting that the loss of RNA editing itself is not the major problem. It's the activation of MDA5 that's the major problem. So what got me really excited though about this whole mechanism and this as it relates to our coronary artery disease is that my colleague Billy Lee, one of our professors of genetics at Stanford, had made an intriguing observation, which is that common genetic variants can actually influence the frequency of RNA editing. So for example, you can have a SNP that would be associated with, say, a lower editing frequency at an editing site. And what was found though was that these editing QTLs, that now we think of it, these are the, rather than expression QTL, actually an editing QTL, were actually associated with numerous common autoinflammatory disorders, including psoriasis, inflammatory bowel disease, rheumatoid arthritis, lupus, but also coronary artery disease. And so what it's suggesting is implicating this axis of ADAR1, where if you have common variants that decrease RNA editing, can actually increase the risk of coronary artery disease. And it's implicating this axis of ADAR1, double-strand RNA, and MDA5 as leading to disease. Intriguingly, a couple years before that, um, Sek Kasrithian's group at the Broad had published that rare loss of function variants in MDA5, IFIH1, was actually associated with a reduced risk of coronary artery disease. And so it's painting this further picture of this being strong genetic evidence to implicate this mechanism and axis underlying a significant portion of coronary artery disease. So what this combination of data had implied was that there is a new paradigm of how we think about ADAR RNA editing in disease, where if you have impaired RNA editing through common genetic variation, you can actually increase coronary artery disease risk. And similarly, if you reduce MDA5, it can actually be protective. But this wasn't a mechanism. This implied strong human genetics evidence, but the question was, through what mechanism does ADAR1 mediate this disease risk? And this was what my work was focused on trying to answer. And so this was mentioned, this was just recently published, but this represents 4 years of effort to try and elucidate this underlying mechanism. And I like to think of this as that we kind of created a new discovery of the role of ADAR1 in RNA editing atherosclerosis with the goal that this hopefully actually creates a new path for an avenue for precision cardiovascular medicine. And so I think I first want to just start this story by asking the question of, well, what cells within the atherosclerotic plaque actually express these RNAs that can activate MDA5? And what's intriguing is that while we can think of it as the thousands of different mRNA transcripts, only a portion of them actually form these long double-stranded RNA structures, and it has to do with their overall sequence, and maybe some just kind of random piece that they form these double-stranded RNA structures, and they require RNA editing. And so for this list, we have, say, we can identify some like 600 of them. We can take this list and we can ask, well, what cells within plaque actually express these immunogenic RNA? And intriguingly, the smooth muscle cell appeared to express these immunogenic RNA at much higher level than other cell types within plaque. And so this kind of points us into the smooth muscle. And I want to take a moment to think about what we understand about the smooth muscle in the development of plaque. And this goes back into decades of what we understand. This is really an old question. Um, I had trained at the University of Washington, which is where kind of luminaries in vascular biology, Elaine Rains and Russell Ross were. And they had described decades ago about this concept that in disease states, the vascular smooth muscle cell can change its phenotype. It can go from more of this contractile type smooth muscle cell into this more what was described as the synthetic, more active smooth muscle cell. But I'd argue that there's been major advances since that time in terms of development of different technologies to ask these questions. So one, we've developed new complex mouse models of atherosclerosis with smooth muscle lineage tracing. We now have a much better understanding of the genomic architecture of coronary disease with hundreds of different genetic loci. And we now have single cell RNA and attack sequencing that allows us to gain better insight into the development of changes in phenotype. And I would want to remind everyone here, which maybe I don't need to given the audience, is that the smooth muscle cell is actually the primary cell type in plaque. For example, this is just some of our data here. If you use an MYH11 driven CRE that can turn on TD tomato, we can lineage trace cells that are derived from smooth muscle origin in the media as it integrates into the plaque, and then what you reveal is this significant engraftment into the plaque of these smooth muscle derived cells. And the Quatermos lab has spent a lot of effort over the last 10 years or so trying to define these cellular phenotypic traits as the plaque develops from the smooth muscle. And our group has suggested a major phenotype that changes, where from the mature smooth muscle cell, there's this activation driven by several different transcription factors that leads to development of a more fibroblast-like smooth muscle cell that we call a fibromyocyte, or we list here just as a FMC. And then this further gives rise to a more chondromyocyte-like that's generating more calcium-rich plaque that we think of as the CMC. And so ultimately, there's this proportion where it's the mature smooth muscle cell ultimately changes its behavior and changes its phenotype that contributes to the development of plaque, and that the factors that govern this transition are important that influence cellular plaque size and stability and other features of plaque. But we could ask, well, how do we know it's important? Well, I think it's intriguing. Several groups have done this now to be able to ask, what do we know from the human genetics data, and what cells actually express these genes that are important in regulating disease risk? And what we have identified is that these cells that phenotypically modulate and invest into the plaque actually retain a significant portion of that genetic risk. And it highlights the idea that the importance of these cell types in mediating disease risk. And so one question we could ask about this as it relates to ADAR and MDA5 and downstream ISG induction is we can say, well, during this phenotypic change, is there any evidence of ISG activation? And what we do see is, so this is taking comprehensive single cell data set with smooth muscle lineage tracing, and this is just smooth muscle cells. So we can compare from week 0 to week 16 in the mouse this phenotypic change. You can see loss of MYH11 and gaining of CMC markers like COL2A1. And we can ask, well, where is there any evidence of activation of ISGs? And we can see this distinct expression of ISG markers with this phenotypic change. We can then look to see if this also occurs within human, and we can take that human atherosclerotic plaque and we can look at the smooth muscle cells, and we can see that phenotypic modulation where you get this loss of MYH11 from here cluster 2 to 1 to 0, and then turning on of FMC, CMC markers like TNF-RSF11B, and then if there's asking as if there is any expression of these ISGs. Again, we see distinct activation of this ISG program, specifically within this cluster 0 as it modulates out to the FMC SMCs. And so this implicates that ISG activation appears to occur in the vascular smooth muscle cell in the development of atherosclerotic plaque, both in human and mouse. Well, we can then ask, well, how much does ADAR1 control RNA editing in human smooth muscle cells? And we can evaluate this by culturing primary human coronary artery smooth muscle cells in a dish. We can then knock down ADAR1 with just siRNA, and we can then, through bulk RNA sequencing, evaluate editing and editing frequency. And what we see is this pretty profound linear relationship between the expression of ADAR and the actual editing frequency. And it really highlights the idea that ADAR1 is the master regulator of RNA editing in smooth muscle cells. And what's intriguing is that when we knock down ADAR1, we see this massive transcriptional change. We see all this activation of all these downstream ISGs, as well as numerous other changes in gene programs. But when we co-delete MDA5, we block almost the majority of this gene expression effect. So in our human data, though, in our human and mouse single cell data, though, we're implying that there's something about this phenotypic transition where they're going from the mature smooth muscle cell into this FMC, CMC phenotype that appears to activate this program. And so it's a question, well, is there something about this phenotypic change? And so we can try and model this we can take our primary smooth muscle cells, we can serum starve them. This is more of like this highly contractile cell type. This is more similar to that SMC cell. And then we can then replete with serum with serum. And this kind of activates more of an FMC CMC like program. And we can repeat this with then deletion of ADAR1. And what we see is with the phenotypic modulation assay, all of a sudden the effect of ADAR1 is dramatically increased in an entirely MDA5 dependent mechanism. So when you co-delete MDA5, you block this almost entirely. But what's particularly intriguing is that when we look at, say, ISG markers, such as ISG15 here, you can see ADAR knockdown, you get this increase of ISG15. It's further increased with serum modulation, and then as co-delete with deletion of IFH1, it entirely blocks this effect. But with other markers, such as key transcription factors that influence phenotypic changes, such as KLF4, EGR1, ATF3, they similarly get turned on, further increased with serum stimulation, and then entirely blocked with MDA5, highlighting that MDA5 itself appears to be regulating these key transcription factors that mediate phenotypic change. And we can look at, say, other markers of, say, that mature contractile phenotype like ACTA2, CNN1, and we can see that in our serum stimulation model, this causes a downregulation of ACTA2 and CNN1, as we would expect. ADAR knockdown itself decreases this, which further decreased with serum stimulation in an MDA5 dependent mechanism, highlighting that activation of MDA5 can directly affect that phenotype. So what's intriguing is that we can then look at the RNA editing frequency across these different cell types, and what we see is just stimulating with serum itself appears to have a subtle drop in RNA editing frequency. And what's intriguing about this is that it highlights that just changing the phenotype itself of the smooth muscle cell can actually lead to decreased RNA editing. So this is, for example, plotting individual RNA editing sites. You can see this overall shift down where you see this lower level of RNA editing in the serum simulated compared to the starved phenotype. We can then look at all these different editing sites when we delete ADAR, and this is also with co-deletion of MDA5. You can see there are all these different editing sites that have very decreased RNA editing frequency. And what's intriguing is that while many of these RNA editing sites are the same for both serum starved and serum stimulated conditions, there are distinct differences as well. And so there are dozens of genes that have distinct editing frequency based on the phenotype of the cell. And what's intriguing is that many of these genes actually overlap with known CAD risk loci across the genome. Okay, so I think what we've Shown here is that ADAR1 and MDA5 appears to play key roles for the SMC phenotype in vitro. But the question is, well, what does ADAR1 in the smooth muscle do in vivo, and can we model that? And so we can try and model that with using an ADAR floxed allele. So with this, we can then express Cre in a tamoxifen-inducible manner through using the MYH11 locus, and we can then do lineage tracing with the ROSA TDT and on an ApoE background. And at 8 weeks age, I can give tamoxifen to induce deletion of ADAR within the vascular smooth muscle. And what we found was that these mice develop a profound illness very quickly. So at roughly 4 days of age, these mice begin to have this hunched over posture. They start rapidly losing weight. They get very sick. And in this first experiment, we had over a roughly 50% mortality by day 14, an effect that was not seen at all for the ADAR het or the control mice. And so it highlights this, the conditional deletion of ADAR1 in smooth muscle causes overt phenotype and death. When we then look at the aorta, what's intriguing is just highlighting kind of how severe the vascular phenotype is. And so you see this evidence of intramural hemorrhage, as well as this, what I kind of describe as more of an adventitial ballooning. It's like from the adventitious side, it starts like ballooning outwards, and you see TDT loss consistent with smooth muscle cell death. And I was then able to do significant single cell RNA sequencing at this 2 week time point. And what you can see is that there was a dramatic transcriptional change with the SMC ADAR knockdown compared to the control. We can see here, this is with the lineage traced smooth muscle cells here. From the control, there's this massive shift outwards. And when we look at expression of genes such as ISG15, kind of a key marker of ISG activation, we see it just kind of turns on throughout the smooth muscle, as well as other cell types. We also see new infiltrating macrophage cells, as well as a small portion of T lymphocytes. And then the fibroblast population changes dramatically with increased SCA1 high expression, implicating a dynamic fibroblast change. And if we then look at the smooth muscle cells, ask, well, what about those markers of phenotypic change? And again, we see that with loss of ADAR, we get this distinct downregulation of key contractile markers like CNN1 and MYH11, consistent with what we saw in vitro. So what's intriguing is that it highlights this idea that the smooth muscle can regulate this dynamic immune response throughout the vessel wall. And so we were trying to get at this question of what's regulating this, say, this new macrophage cell infiltration. And we can get at this through cell receptor ligand interaction modeling through CellChat. And what we had identified was that CCL signaling is the top interacting family following ADAR knockdown, and particularly CCL5, CCR5 was the strongest. And what you can see is that with the clusters that are associated with SMC ADAR knockdown have key outgoing signals of CCL5 with key incoming signals of the infiltrating macrophage cell population and T lymphocytes for CCR5. And what's impressive is that you can just look at this on feature plot. You can see that the smooth muscle with ADAR knockdown is like a light switch of CCL5 getting turned on and CCR5 being highly expressed within the infiltrating macrophage cells, suggesting that following ADAR loss, this distinct CCL5, CCR5 might be an important axis for mediating this inflammatory cell infiltration. But so the question is, well, can inhibition of MDA5 block this effect? Our in vitro data is pretty suggestive that MDA5 is a key regulator. There are other double-strand RNA sensors, including a RIG-I. And so you could ask, well, maybe deletion of MDA5 wouldn't block this effect. And what we found, though, was that with simply haploinsufficiency of MDA5, gene symbol IFIH1, that you completely blocked the phenotype, that there was no mortality seen in these mice, and there was really effectively no even significant weight loss. Appeared entirely protective with IFIH1 deletion. And then I did additional single cell RNA sequencing with the double knockout. And I think this is what's particularly important, and I think is helpful for us just to think about here, is that we show that with ADAR knockdown, there's this massive transcriptional change. But with the double knockout, with single cell RNA sequencing, the cells almost fall back entirely onto the control. And so if we look at G activation here, for example, it turns on and then it's off and it goes right back. And what it highlights is that this group still has effectively lost RNA editing within the vascular smooth muscle, but the transcriptional change appears very modest. And I think that's an important finding, which is that it highlights is that despite loss of RNA editing itself with ADAR knockdown, if you don't have MDA5, the downstream effects are very, very minimal. And so what we've shown here is that MDA5 is required for mortality phenotype with SMC ADAR knockout, and that you can protect against this. And it creating this idea that with the SMC ADAR loss, we get this high mortality, loss of vascular integrity, and this is a rescued phenotype with MDA5 knockout. But I started talking about atherosclerosis, and of course, this is not atherosclerosis. This is like a severe short-term model. So the question is, does impaired SMC-specific RNA editing accelerate atherosclerosis? And to evaluate this, I wanted to utilize the SMC ADAR heterozygote model. And so in this model, there's these mice appear entirely normal. They don't have any, they don't appear to have any clear phenotype that we can identify. And so the question is, well, is there something about atherosclerosis that may change this? And so by using a 16-week high-fat diet model with our ApoE background, following 16 weeks, we can then do single-cell RNA sequencing and histology. And maybe I'll first just show the single cell data. And I think what's exciting about this is in the control group, we're kind of seeing the cells that we would expect. So here's the SMC lineage trace cells, the cells that we call smooth muscle, the SMC, FMC, and then CMC clusters. We see, as we expect, fibroblast population, macrophage population, some T cells, endothelial cells. This is our general, fairly typical control population. And then with the SMC het, all of a sudden, ADAR het, we also see this distinct new cell population that lies right between the SMC and FMC populations. And when we then evaluate what is the cell type, we see that it's marked entirely by ISGs. And there's this distinct expression of ISGs at this phenotypic change, which is quite intriguing. So we can take the smooth muscle cells, we can then subset on them, and we can really highlight this distinct difference with this SMC ISG cluster that I'm calling. And we can then ask, well, what about those different cellular makeup? So we know there's the SMC, the FMC, and the CMC. And what we identified was that with the ADAR het, we see that increase of the SMC ISG cluster, but there actually is a 40% increase in the CMC clusters. And it highlights this that there may be a relationship between the SMC ISG and ultimately the CMCs. On histology, what we see is that plaque is increased in size at the root. So we see this increased plaque size. And probably importantly, we see this pretty clear, distinct decrease in media thickness in this media area. And I think this highlights, and this was then suggested with evaluation of the TDT within the plaque, is that there's more migration of cells out of the media into the plaque, and you see decreased media thickness. When we look at RNAscope across the plaque, you can see that there's just in the control population, and this is an important point too, is that in the control, we can see ISG15 expression within the media as well as at the base of the plaque and kind of up close towards the cap. And then with the ADAR het, it's just increased significantly, and particularly, arguably more at the base of the plaque. When we do stain for pharyngeal blue as a marker of alkaline phosphatase and calcification, we see there's this distinct increase in vascular calcification, which is fitting with the increase in the chondromyocyte population, which actually also is fitting with what has been reported clinically, which is that patients who have loss of ADAR or gain of function of MDA5 have a distinct increase in vascular calcification at a very young age. So this is a cross-section of a CT of a young patient who has severe aortic calcification. And so the question is, well, how do cells go from this mature smooth muscle cell into this chondromyocyte? And these are lineage trace cells, so we know that they're going from one to the other. Our group has previously reported this transition from the smooth muscle more into a fibromyocyte-like and then into the chondromyocyte. But by performing lineage trajectory analysis using Slingshot, what was intriguing is that we identified not just one trajectory from the SMC to CMC, but actually two. And this second trajectory is interesting because it specifically involves the ISG clusters. And it highlights this idea that this ISG, what I term as ISG dependent trajectory, may be distinct from a different ISG independent trajectory. We can then look at this transition point and look at all the different clusters, and we can ask what genes do they appear to express. And you can see that there's particularly activating numerous different ISGs, but there do appear to be key regulatory nodes, including IRF7, STAT1, PIK3, and others that appear to regulate this phenotypic change. So this was, I think, a really important experiment that took a lot of time and effort, but I think it's helpful for us to show here, is that we had shown that the ADAR het mice appear to have this distinct activation of this ISG cluster as well as increased chondromyocyte. And we had previously shown with homozygous deletion of MDA5 or heterozygous deletion of MDA5 that we could protect against complete loss of ADAR. But the question was, if we just simply have MDA5 heterozygosity, will that protect against this athero-phenotype in the SMC ADAR het model? And what we had found was that the answer was yes. And so with this is the control. With the ADAR het, you see this distinct activation of the ISGs as well as increase in the CMC. And then with the double knockout or with the ADAR het and the MDA5 het, you completely prevent the SMC ISG cluster. But what was intriguing is that not only was this in the het, there was the 40% increase in CMCs, there was actually a 30% decrease in CMCs compared to the control in the MDA5 het background. And when we pull out the differentially expressed genes and we can plot them on a heat map, what's interesting is that you can say control group in the middle here, this with the ADAR het, there's all these genes that have increased expression. Maybe that's what we would expect. But with the MDA5 het, not only are these genes not increased in expression, the expression actually appears to be even lower than that in the control group, which I think is an important observation. I can show some specific examples here. So if we look at ISGs, the ISG expression in the control is relatively modest. It goes up in the ADAR het group and then is down to effectively zero in the MDA5 het double group. But if we look at markers of the CMC FMC transition, such as things like VCAM1, Lumacan, FN1, TNF-RSF11B, they're expressed at a reasonable level in the control. They go up in the ADAR het, and then they get decreased to even lower than control in the ADAR het with MDA5 het. And this is pretty strong evidence to suggest that MDA5 is being activated even in the control group, to suggest that deletion is maybe protective. So the question is, well, is this mechanism involved in larger human populations? And so we tried to evaluate this. And so we had developed our collaboration with the Athero Express cohort network, which I'm very grateful for, where in this evaluation of 1,093 carotid endarterectomy samples that were paired with RNA sequencing, histology, and clinical history, we asked arguably a relatively simple question, which is that one, do we see that expression of ISGs match with or have any relationship to expression of phenotypic transition? So we looked at expression of things such as Lumacan and Haplin, that's strong markers of phenotypic change in human atherosclerosis, and then looked at this in relation to ISGs. And what we found was this distinct linear relationship with very strong significance. And here we're just plotting what the negative log p-value is for the relationship of Lumacan, linear relationship of ISGs and Lumacan, and you can see these very strong relationships. But what was particularly intriguing to me was that the expression of some of these ISGs, in particular IFIH1, which is MDA5, has a very strong relationship to plaque vulnerability. So now you have IFIH1 relating to plaque vulnerability with a negative log p-value of like 1 times 10 to the negative 130. Similarly, you see the strong relationship to plaque calcification, which is fitting. And so I think what we've done here is I think we've started from the human genetics to be able to create a more comprehensive mechanism and understanding of the vascular smooth muscle and activation MDA5, where that deficient RNA editing and MDA5 activation appears to be a causal mechanism of coronary artery disease. And when we think about how this can be applied therapeutically, we actually have the ability to sequence patients and actually identify patients who would be predicted to be a double-strand burden high, as in these are the patients who have reduced RNA editing, where this mechanism may be more at play. And we have an active program to develop small molecule inhibitors of MDA5 with a goal for a future clinical trial to select for those with high double-strand RNA burden to randomize MDA5 inhibitors. Okay, that was the start from there from the overall scientific story. There's a lot that I did not talk about this in the paper. Admittedly, the paper is relatively large. Um, I was also asked to talk a little bit about the publishing of this. Is that if that's okay, maybe I could just go through this a little bit. I was thinking when I was asked to talk a little bit about what the publishing experience was like, I was a little bit hesitant. I was just thinking about like what could I maybe talk about that would be that would be reasonable to discuss. But first, I want to say that I am enormously appreciative of all the reviewers who put in significant effort to this paper. It's clear the reviewers put in hours and hours of effort into reading and reviewing and putting in detailed suggestions. And it did improve the paper tremendously during the review process. So I'm very appreciative of that. I also want to say that I'm extremely proud of how the paper turned out. And I think for all of us who are working in this field, like, boy, all this research is hard, right? Everything is hard. And so we do need to take effort to kind of celebrate when this stuff comes out. And I do think Nature Cardiovascular Research is an outstanding journal to publish in. Um, what I would say is that in my experience, there appears to be a distinct challenge when you're publishing work that spans across complex yet different disciplines. And I think this might be an example of this, where you have RNA editing, which is like distinct biology of ADAR biology and RNA editing. You have complex genetics. Then you also have smooth muscle biology and atherosclerosis. And arguably, these are complex and distinct fields. And I would say that with the review process, I would say at times reviews may reflect appropriate and on-comment targets in one area, say like RNA editing, where similarly other comments may feel less relevant or perhaps not on target, for example, like in atherosclerosis. And I think it just highlights this challenge because sometimes this discrepancy can lead to challenges in how the paper is reviewed. And I do think that there's this fundamental challenge when trying to publish this interdisciplinary work, which is that by nature, reviewers may have expertise in one domain while lack expertise in another, which is what you would expect. And I think this is where a really good editor appears to be crucial to be able to understand and modify expectations for reviews. Um, I would say that this was a long road to publish. So first, this paper was first put together in March of 2024. And so that time, the paper had the human single cell data, most of the HCASM data, the short-term homozygous SMC8R1 knockout, the 16-week ADAR het athero data, and then also the ADAR knockout with MDA5 double knockout data, but I didn't have the single cell data from that yet. But I think overall the story was still there. It was implicating MDA5 and the increased athero. Um, we had submitted to Nature as just like a reach journal. They had initially published the RNA editing work before, so it was just thought about, well, maybe it could be like a follow-on to that. And they suggested transfer to Nature Cardiovascular Research. At the time, I had this idea in my head that maybe it would be great to have it more in like a general journal. So I wanted to, I sent to Science Translational Medicine. It went out to review. I would say, long story short, 8 months there, three revisions, additional in vitro experiments, additional in vivo data, all the new single cell data. I had two reviewers saying ready to publish, and then another reviewer saying no. Fourth reviewer had new concerns. Ultimately, did not land there. So that was tough. So then it was October of 2024 that I had submitted the paper to the AHA Basic Cardiovascular Sciences for the CATS Prize, where I was a finalist. And then as part of that, Circulation invited me to submit the paper for an expedited review. And I was thinking, okay, this is going to be great. This would be perfect. That did not work either. So then sent it to Nature Cardiovascular Research. And at this point, I did now have the ADAR het with MDA5 het, athero, and single cell data. So I had included that in a reformatted the paper. Ultimately, the reviews were positive, but there are ongoing requests for more animal data, more in vitro data. Now there's a request for it to be shown in a larger human data set, which led to the Athero Express. What we did was we met with the editor and were able to build a plan to defer the in vivo experiments. That was going to take me like a year and a half to do. Could do the in vitro experiments and plan to build a human collaboration data set. And so this was my first experience with this, where the editor then reached out to the reviewers by email to share with them like the preliminary response with the plan and to see if they were okay with that. And they all agreed that that was reasonable, and then was able to complete that and then have it accepted in August. So this was a year and a half since first submission. So this was a long road. But I will say that this paper significantly improved. So if I look at what it was at first compared to now, it is very different. And while I think it is probably true to say that the overall main takeaway and theme of the paper did not change from the first submission to publication, but the manuscript itself improved and it laid the groundwork for all these other, like the new in vivo data, new in vitro, and human data was substantial. Um, and the paper is admittedly massive. Like, there's like 8 manuscript figures, 10 extended data figures, 10 supplemental figures, dozens of single cell and bulk RNA-seq data sets. Um, I think there's an ongoing reasonable question as to how it could be packaged into multiple papers. And my initial goal was to have the MDA5 knockout story as a second paper, but that obviously didn't happen. Um, but I do think that this paper can perhaps stand as more of like kind of a landmark in like kind of the RNA editing vascular biology field, and it can also serve as a strong start to my lab. So I'm in this transition period where I'm now starting my lab and moving into an independent track. Um, and so I think with that, I'll just say thanks to this was my advisor, long-term advisor, Tom Quatermos, collaborators such as Paul Chang, Billy Lee in genetics, and Chin Lee, who's now over in genetics at Penn, Sander van der Laan at the UMC Utrecht, who had helped with Clint Miller, who'd been fundamental with the Athro Express data. And if there are questions or if you're interested in thinking about whether or not like a postdoc, for example, on RNA editing at Stanford, please let me know.
Brilliant. Thank you so much, Chad, for this wonderful presentation. And before we jump into the Q&A session, I like to stress something, like this. Journal clubs are meant to give space for like ECRs and like to help the understanding how good science works. And I really like you sharing your experience with the review process because, it takes a great scientific question and great science to publish a good paper, but also there's much more work, to be done there. And um, if anyone has a question, please feel free to come forward on or to type it into the Q&A in the chat. We can ask. I can see a hand raised. Miran, please come in, ask your question.
Yeah, thanks so much, Fernando. Chad, I think this is amazing. I mean, the journey of this paper is for award itself. I think I congratulate you for your persistency to actually came up with all the solutions and improve the paper that in the beginning was very interesting story, and then how you pick it up and how the story progressed was fascinating. I was wondering because at some point you have been mentioning that you are working on the selective inhibitors of this MDA5, if I mention it correctly. So is this something that is relatively selective to only plaques or vascular smooth muscle cells, or this is like ubiquitously expressed across different tissues? And how would you see this progress more towards something that is personalized use in clinics? Is it something that you need to deliver on site? Are there any adverse systematic effects?
Yeah, no, I think it's fair that we can actually maybe what I'll do here is I actually have a couple more slides. Actually, I wonder if I could figure out how to get to these slides. Um, there's let me actually see if I can pull this up. It'd be easier for me to show this in a couple of prepared slides that I had. And I see there's a question in the chat about the modality of how well it's going to or antiviral response and question about that. Okay. So I think this relates to this in general. So if we um, think about MDA5 as a therapeutic target, one, we have like really strong human genetic evidence of it being causal in disease. which is a really important place for us to start about it. But to the question of MDA5 is expressed across all sorts of different tissues, and the expression, as we've shown, can increase with distinct change. So, for example, the expression in plaque appears to go up in MDA5. MDA5 expression appears to go up in the development of plaque. But there's a reasonable question about its targeting in terms of playing a role in antiviral immune response. And so that's a reasonable question. But we have some data that may actually provide some evidence to say that it's feasible. And one is that we oftentimes start with this idea of what do we have from human genetics evidence to say that inhibition would be deleterious. And there's this concept of what we call genetic constraint. So in genetic constraint, it basically implies this idea that a rare loss of function variant of that gene would not be reflected in the population. So we could actually say, well, what would be the expected amount of rare loss of function variants versus what do we actually observe? And this observed to expected loss of function variant ratio is quite important. So for example, the observed to expected loss of function variant ratio for ADAR is like 0.3. For another gene that we know and love, like PCSK9 for inhibition for lipid traits, is like 1.0. Interestingly, for ADAR1, it actually is like 1.17. Like it just nearly misses significance for actually being selected for, which is pretty strong evidence to suggest against it having constraint. So what's also important is that when we think about viral sensing, while MDA5 is an important one, it's actually a minor one for viral sensing. RIG-I is like the dominant RNA sensor for multiple different viral pathogens. And several other viruses actually overlap where RIG-I can sense as well as MDA5 can sense. So MDA5 actually only senses a small minority of viral pathogens. And what's interesting is so picornavirus is one, like encephalomyocarditis virus is one. But what's interesting is that there was actually some studies going back like 20 years ago where they asked like, well, what is the effect of MDA5 knockout with say following treatment of a virus that requires MDA5 like encephalomyocarditis virus? And what they found was that while knockout mice, if you look say at their interferon response and their ability to clear the virus, while the knockout mice had impaired interferon response and succumbed to the viral infection, the heterozygote mice actually had a perfectly normal interferon response and actually cleared the virus. And so in my mind, the way I think of it is that this question of like therapeutic index is actually quite intriguing, is that if we map this as say like the percentage of inhibition of MDA5 that's in overall RNA sensing, is that there appears to be two different curves that for endogenous RNA sensing, inhibition of MDA5 appears to basically block endogenous RNA sensing with just like simply 50% loss. But the viral RNA sensing is preserved, but it implies that there's this range in between where if you get partial inhibition of MDA5, that you can actually have preserved viral RNA sensing while inhibit the endogenous RNA sensing, which we think of as driving the pathogenic effect. But from like a therapeutic strategy standpoint, the goal is to actually have like a small molecule inhibitor that would actually inhibit throughout tissues, and in part because it's a common mechanism that appears to also be important for things like lupus and psoriasis and other things. So there's other disease indications.
Thank you so much. If while anyone else has time to think about their questions, actually, I think mine ties to this as well. So looking at what you just explained, which was very nice to discriminate between the endogenous DNA, double S, RNA sensing and the viral one, and also looking at your sequencing data, you were mentioning that actually the MDA5 heterogeneous knockout has a lower level compared to controls of this kind of CMC transitioning capacity, let's mention.
Yeah.
So taking these two things in mind, what is your design for like the dysfunctional screening to identify MDA5 inhibitors? Because I guess there is something like too much NDA5 inhibition. You know what I mean? Like, what is your target when you look at these potential molecules? Because you are not likely to want to target the ones with the highest inhibition score, because I don't know, like too much could be like detrimental in a way in the atero context, at least. I'm thinking based on your data.
Well, No, I think like what we would say is that the endogenous RNA sensing by MDA5 appears to have an adverse effect. And I don't know if we would say that too much inhibition of MDA5 would cause a problem. I guess I would say too much inhibition of MDA5 would be a problem from like a viral infection standpoint, like another standpoint. But if we're going to hit that partial inhibition, our group is actually in with Billy Lee, we've actually already conducted a large small molecule screen of two different parts, and we actually have
Okay.
compounds that are now prioritized and working with our Stanford ChemH team in terms of optimizing from there. So
Brilliant.
we're kind of moving towards that direction, which is exciting.
Indeed, it's super exciting. All these functional screenings, finding repurposing drugs and so on can be very, very rewarding as well. So I
Yeah.
think we have a question from Melody.
Yes, thank you for a very exciting presentation and amazing work. I might go back to the more smooth muscle cell part of the study, and I thought it was really interesting how you described that you found maybe like a new way to the CMC
Yeah.
trajectory. So
Mhm.
what do you think makes the smooth muscle cells go through an ISG versus a non-ISG trajectory? And also, I thought the data on the KLF4 interaction was quite interesting. Are you
Yeah.
planning to pursue this a bit further to see how this
Yeah.
modulation can happen?
Yeah, actually, and I actually have I've actually developed a new mouse model trying to evaluate this. So I think one thing that's the intriguing from that data is that it implies that there are cells that can transition into like an ISG activation, and whether or not that's just the ISG activation that pushes them in this direction or interacting with other transcription factors, but they'll turn on ISGs, and then our data implies that they then turn it off, as in
Mhm.
like when they move to the CMCs. So, for example, like the CMCs within the plaque don't appear to overlap with cells that are expressing ISGs. Like they're separate cells, but then it appears they're contributing to that. So it implies that there's cells can turn on like an ISG program and then subsequently turn it off
Mhm.
as they transition. And so the question may be, well, how can we actually map those cells and truly understand how one cell becomes another?
Okay.
And so what I've done is developed an ISG15 DRE recombinase that's tamoxifen sensitive with a DRE responsive TD tomato. And so what it allows me is that through an orthogonal approach with a DRE, I can still use, say, a CRE to say in haploinsufficiency delete ADAR, but then label any cell that turns on ISG15 permanently. And so
Okay.
within the smooth muscle cells, I can then see how cells, TDT positive cells, can contribute into the CMC, for example, is that we can then map those cells in a much more high resolution to understand how those cells are then contributing to the plaque. Because I think that's an ongoing question is like, how do cells that activate ISGs truly contribute into the plaque?
Mhm.
Um, but I think what's interesting about this is that my hypothesis is, and our data implies that these cells just become CMCs. Like they actually transition, they turn off their program, and they become CMCs. Like it's like they've changed their fate commitment through activating of these programs. But if it's, for example, if we do this lineage tracing study, and now I have these mice and we're um, now starting like on a high fat diet and everything, but like it's possible that they don't, and if they don't, then it'll be a question of, well, what cell-cell interactions are actually driving the increase in CMC? And I think that's going to be another question. Because I think classically there's been this description of like cells kind of come up along the plaque on the cap and then they kind of go down into the core of the plaque to generate. But I think we're also implying that there might be just this path where it just kind of goes straight up. It's like from the media up into the base of the plaque to form increased
Different
calcium.
trajectories
Yeah.
from every
Yeah.
location. Exactly. Yeah. Yeah.
And
That's
so,
very
and I think
interesting.
that's what's intriguing is that we can think of it as like different trajectories and we can better map that.
Mm.
And I think that'll be important.
That's really nice. Thank you.
Brilliant. I think we have time for one final question, if anyone has a burning one. Otherwise, I think Chad will be glad to keep in touch if anyone wants to follow up with other questions. If not, we are coming close to the end of this hour, so I think I can pass it on to you, Mel, to wrap up the session.
Perfect. Thank you for a very amazing presentation, and thank you for agreeing to take part in the Journal Club. I hope that this gave some a platform for people to be, contacting you, and then maybe sparking more collaboration. We will be keeping up with your research, Chad.
Yeah.
And I can close the session and say thank you for the presentation, and thanks for a very interesting discussion.
Great. Thanks so much. I appreciate you guys all coming.